So it's our pleasure, it's my pleasure to introduce Katie Morrison here. She's from University of Northern Colorado, although now she's visiting Penn State for a sabbatical, an extended sabbatical. And I met her like so many years ago, like when we both were PhD students in coding theory, and now we seem to do a different path in research. It's going to be a pleasure to hear her. The talk is Emergent Sequences from Neural Networks.
Thank you, Felice, and thank you to the organizers for having me. So I want to just start with a little bit of the sort of neuroscience questions that are motivating this work, and then I'll get into more of the actual modeling. And I will warn you now that the model itself is continuous and doesn't feel like it exactly fits in this realm of things. But what we're going to end up looking at it is features of the graph that are defining it. And so we really will eventually land in discrete world, I promise. But so the main problems that are motivating this work is an attempt to understand sequences in the nervous system. So just as an initial example, so in the hippocampus, there are these particular neurons called place cells that have little convex regions in space that when you enter into that region, it will cause that neuron to fire. Okay? And so these cells are tracking or used for spatial navigation. And initially people thought that it was really just sort of static attractors that were representing the animal's position at any given point in time. But with higher resolution recording technology now, they've actually been able to see that you're getting a sequence of neural firing that's encoding the particular position. And here's just an example as a rat is running down a linear track. And one of the things that's particularly interesting is, okay, so we get these sequences that are evoked in response to the rat actually running, but we also get sequences even in the absence of any external input like actually running, okay? So when the animal is at rest or during REM sleep, we get these sharp wave ripple events. And during those events, you again get sequences of neural activity. In this case, you're getting a sequence that's really similar to the sequence that we saw as the rat was running down the linear track. But in other cases, so this is data from Eva Pasalkova's lab, who's a research collaborator of ours, or an experimentalist collaborator of ours. So here are example neural sequences, and again, that are coming during these sharp wave ripple events, so while the animal is at rest. And you can get, let's do this little pointy thing. So in these sequences, you're getting the same type of activity as this forward running down the track. So it is sort of a replay kind of sequence. But interestingly, you also get backwards sequences of activity, even though the rat has never run backwards down that track. Okay? And you get other sequences of activity that don't correspond necessarily to a linear progression one way or the other. Yeah.
Again, what was this, the stimulus, something entered a space? What was that thing?
Sorry. There is not an explicit sensory stimulus for this. What it is somehow, even though hippocampus does not have sensory input, so it's still unclear, it's the rat's position in space, or a person's position in their surrounding environment. So here, what these little color-coded blocks are showing are this particular region of the linear track causes that particular neuron to fire. Whenever the rat is in that region, that corresponding neuron will fire.
And it correlates to the color?
Oh, the color is what we're using to represent that data here. Yes, but in general, there is a correlation between an animal's position in space and a particular neuron's firing.
A change in space? A change in position, in other words, an acceleration?
It is not, in this case, it's not about acceleration. It's not about speed or anything like that. It's literally about actual position at any given point in time in your 2D environment.
So if the animal moves very slowly in space, that will stimulate it very slowly?
Yes.
And you don't know the mechanism?
We don't know the mechanism. No, because there isn't direct sensory input, and somehow hippocampus is acting like a sort of GPS system in some sense, without getting any of these. Like, it can be affected by particular like cues in the environment, can help align things. You still get place cells even in animals that are blind. And you get place cells, you get three-dimensional place cells in bats that are flying. You get, yes, they are not specific to, they are not necessarily like sight informed. Yeah. But key idea is, I mean, one of the things that they think is shaping the emergence of these sequences is something about the connectivity structure of the network in hippocampus. Okay, so as the rat has learned this forward trajectory, that has informed the structure of the network or shaped the structure of the network in such a way that even in the absence of an explicit stimulus like this, you're still getting this kind of activity. They also see neural sequences in cortex, so transitions between collections of neurons firing together. And you can see these sequences both in response to an actual stimulus, but also spontaneously. So in the absence of any external input from a stimulus, you still get spontaneous transitions between sets of neurons firing. Lastly, I just want to also mention that you can see sequences of neural activity from central pattern generators. So here are just two examples of collections of neurons that control locomotion of the crayfish and then of the lamprey. And you can see the sequential firing of those neurons that corresponds to like the swimming of the legs in the crayfish. Okay? And so one of the sort of main mechanisms that people understand as leading to these sequences of neural activity is that of disinhibition. So the idea is that there is, in some sense, competition between these neurons over who should be, or sort of dampening the overall firing activity of these neurons. And then as one neuron peaks, right, it tends to dampen the other neurons more. As its firing rate decays, there's enough disinhibition to enable another neuron to start firing. Okay? And so central pattern generators in particular are an inhibition-dominated regime. We have a lot of inhibitory neurons that are enabling that competition. So the main question motivating our work is, given a recurrent network, so something that doesn't have just an obvious feed-forward kind of structure to it, but can have cycles in your network, can we predict the sequences of activity that will emerge? Okay? And in particular, we're interested in how the connectivity of the network shapes that sequential activity. So one of the things that has been a big push of the Obama Brain Initiative and a lot of other Blue Brain Project in Europe has been to try to develop these connectomes, so full connectivity graphs, essentially, of brains of different animals. So they accomplished this for itty bitty worm, C. elegans, a while ago. They have done it for zebrafish larvae now, but there's sort of, even the zebrafish is already 15,000 neurons, okay? And you have all of the connections between these neurons via synapses, or an understanding of what all of these connections are. And so the experimentalists want to know, okay, we have this data now, what do we do with it? How do we understand what is coming out of this connectivity data? What does it tell us about what we should expect for the neural activity?
How varied are the graphs from zebrafish to zebrafish?
So I don't know that offhand. We have just started collaborating literally in the last month with a zebrafish person. And so I don't know at this point, but I could get back to you on how varied that was. I mean, C. elegans, the worm that had only 300 neurons, that I am more familiar with. And there isn't a lot of variability across those in terms of the hardwired connections of the synapses, but that doesn't tell you anything inherently about the strength of those connections. And that's where people see learning as taking place a lot. And so it's not clear that knowing the connectivity necessarily tells you that you know sort of everything that can be stored in that brain. But one of the things in particular that experimentalists have been asking for is, okay, what kind of graph measures should we be using to look at this connectome data, right? So, I mean, you can look at things like, and they have looked at things like small worldness or hubs, or even just like degree sequences and things like this, right? But it's not clear that measures like that, sort of taken straight out of the box from other network science areas, will necessarily be informative in this context. Mostly just because neuron firing has inherent nonlinearities to it, whereas all of these typical network science measures have come from studying linear systems, right? And so one of the questions here is, we want to look for what we're calling sort of dynamically relevant motifs, where what we mean by that is motifs or subgraphs that, based on some model that is neuroscience inspired, really should tell us something about sequential activity that will result. Okay? And then one hope from that would be to even be able to tell you something like specific order of neural firing in sequences based on identifying these motifs. So again, our approach is going to be to develop a mathematical theory in a simple model, but we are going to restrict to a simple model that is explicitly sort of neuroscience inspired, so that we are getting things that should be dynamically relevant. And then we are going to look at inhibition-dominated networks, and again, recurrent networks specifically, not just feed-forward structures. And then our goal is going to be to analyze connectivity graphs for motifs that produce these emergent sequences. So the network structure that we're considering is threshold linear networks, which have a relatively long history in neuroscience. They're also used in deep learning. The threshold nonlinearity is used in deep learning, if you're familiar with that. So the key idea is just that we're associating a firing rate variable xi to our ith node in our network. And then we have a weighted directed graph. And so the rate of change of the firing rate is going to be equal to negative of the firing rate. So this is a leak term that just guarantees that in the absence of any synaptic inputs, the firing rate would decay to 0. Okay? And then we're summing all of the weighted synaptic inputs from these neighboring neurons, plus this external input theta. And because we want to understand these internally generated or emergent dynamics, that theta is going to be constant across all of our neurons. So we're not thinking about tracking a changing stimulus. We're thinking about what's coming from the network itself. And then this last term is inside this threshold nonlinearity, which is just 0 if the inputs are negative, and otherwise it's the identity function. And so with this set of just piecewise linear is our nonlinearity, so it's rather simple. But this nonlinearity is actually sufficient to get us the full range of nonlinear dynamics. So we can see multi-stability, or multiple stable fixed points coexisting in a network. We can see limit cycles, chaos, quasi-periodic attractors, and all of these types of behavior can arise for an effectively inhibitory network W. So what I mean by effectively inhibitory is that all of those synaptic weights are going to be negative. Okay? And I'll give you a biological inspiration for that in a little bit. But so just in case you are not as familiar with all of the sort of nonlinear dynamic attractors, here is an example network that produces, just based on changing the initial conditions of the network, we can get 2 stable fixed points, we can get a limit cycle, and we can also get a quasi-periodic attractor. Or sorry, a chaotic attractor in this case. And all of those can emerge from that same network without changing parameters, just based on what neurons you're initializing the system at. Okay. So as I was saying, we're really building off of sort of a long history of results of using these kinds of networks in neuroscience, but typically sort of inspired by the Hopfield model, which was this attractor network where the connection matrices were all symmetric in order to make the math work out nicely. The main focus of study has been on symmetric matrices. And the problem with symmetric connection strength matrices, well, they are great for giving you stable fixed points, but that is all that they will give you. In particular, you cannot get a sequence of activity if the connection strengths are the same in both directions, right? There's never sort of a particular direction of flow. And so in this past work, we were just getting static attractors or stable fixed points, but our goal is to be able to generate rhythms or sequences and understand the source of that. So one of the things that we're going to focus on in particular is the role of connectivity in shaping these dynamics. And so for the moment, we're going to specialize to this class of threshold linear networks that we've coined combinatorial threshold linear networks, where the connectivity matrix really is fully specified by an underlying directed graph. Okay? So here, what we are thinking of is that there are excitatory neurons, which are these little triangles, and they are surrounded by a sea of inhibitory neurons, but it's general, global, non-specific inhibition. Okay? So really, the direct influence of each excitatory neuron on each other is through these excitatory synapses between them. But there is also an indirect influence that every neuron is competing with every other neuron because of this global inhibition. So when an excitatory guy fires, he makes all of the inhibitory guys around him also fire, which shuts everybody up, except in the case of this direct excitatory connection. So the idea is our graph is going to represent our excitatory neurons, and the edges are going to be less, or like weaker inhibition. So this background inhibition added together with an excitatory connection. So it will always get a weight of -1 plus epsilon in the case that we have an edge. And in the absence of an edge between two nodes, the only way in which they are influencing each other is via that background global inhibition. And we're giving that a -1 minus delta value. Okay? And here, this comparison to -1 is just because we've implicitly set our time constant to be -1. So it's just whether you're inhibiting the other neuron more or less than you're inhibiting yourself.
So the inhibitory neurons are doing basically collision avoidance.
In some sense, yeah, and they're bounding your activity, so you're not going to get runaway activation of things as well. Because you can still have co-firing within this network, so it's not completely only winner take all, but it is preventing runaway activation. Okay, and so in this case, our graph is precisely prescribing our connectivity matrix, other than just a few parameters. Our delta is the strength of the strong inhibition, epsilon tells us about the weak inhibition, and then theta was this constant external input. And because our network is fully inhibitory, that external input has to be positive in order to get our dynamics to flow. So again, the graph encodes this pattern of weak and strong inhibition, and you can think about a sort of generalized winner-take-all network, right? And for fixed parameters, right, only the graph is changing. And so this is what is isolating the role of connectivity, right? The only difference between things throughout this work is going to just be changes in the graph. Okay. So we want to know to what extent, when we restrict to this model, is graph structure really controlling dynamics, right? And the first hint that we had at that was this first theorem that we were able to prove, which states that if your graph is oriented, so there are no bidirectional edges, and it has no sinks, so every node has out degree at least one, then we can prove that the network has no stable fixed points. And furthermore, because it's inhibition dominated, the activity is bounded, and so the dynamics are guaranteed to be oscillatory or chaotic. Okay? So we're going to see this type of sequential behavior. And one of the nice things, even though this is an inhibition-dominated regime, right, activity really is flowing along the arrows. Okay? And so even though it's not an excitatory connection there, actually, like it's still effectively inhibitory, the mechanism of disinhibition is allowing the activity to follow the arrows. And so we really are getting a nice sequential activity in this limit cycle corresponding to that free cycle. So here's just the simplest example, the smallest oriented graph with no sinks. Here is another oriented graph with no sinks, but really here I want to talk about sort of the bigger problem that we're interested in. Right? So this particular network, as it turns out, has a global attractor appears to have a global attractor for the parameter values that we were looking at. And in this attractor, we always get this sequence of neural firing, 1, 7, 6, 3, 4, little 5. Okay. And so it seems to be picking out a particular cycle in the graph, of which there are many cycles in this graph, right? And so what is it that's special about that cycle? Why is that the one being picked out? And how can we figure that out? In particular, it's not something that's like another thing about this sequence that I should point out is neuron 2 actually dies. Okay? So neuron 2 is not participating in this attractor. No matter what my initial conditions are, even if I started out fully, the only neuron that is active in my initial condition is neuron 2, I will still settle into this attractor and neuron 2 will die. And that's despite the fact that neuron 2 has a relatively high in degree of 3, right? Whereas like the high firing node 3 only has in degree 2. Okay? So it's not something that we could predict from just local features like our degree sequence. And so we really want to understand what are the relevant graph structures that are shaping these dynamics. So as I mentioned before, one of the things that experimentalists have been asking for is, okay, can you identify motifs for us? And when those motifs are going to produce interesting attractors, and how we would know from looking at this connectome that that would be true. So just as a sort of simple kind of motif that we might look for, right, we could think about a 3 cycle, which we know that in isolation at least produces this limit cycle dynamics. So what happens when we look at 3 cycles embedded in larger networks? Okay? So here we have a 3 cycle 2, 3, 5, and we get a corresponding limit cycle. Right? And the high firing nodes really are matching and sort of very similar looking to the original 3 cycle. But we also pick up low firing from nodes 4 and 1. Okay? But interestingly, there is another 3 cycle in this graph, 1, 4, 5, and yet there is no corresponding limit cycle for that. Okay? So the question is, why did a 3 cycle produce a limit cycle in one case for the 2, 3, 5, but not in the other case? Yeah?
The weights are fixed in these examples?
Yes, the weights are constant, are all fixed. The -1 plus epsilon for the edges, -1 minus delta for the non-edges. Yep.
In the previous graph, it's 1, 7, 6, 3, 4, 5, 1.
Yes.
So that creates a symmetry where 2 is the focus of the symmetry, right?
Sort of. I will come back to this at the end, and you will see there is symmetry involved here. But I will come back to that in a little bit. Good catch, though. Okay, But so here we want to know what was the difference between these 3 cycles. Why did one of them give us an attractor and not the other? Right. Here's another example network that has 3 cycles in it. But again, 1, 4, 5 does not give us a corresponding limit cycle. And so again, what is it about the embedding of that 3 cycle that is shaping the attractor? Okay. So one thing, so we say that the 3 cycle attractor survives to the full network when we see a limit cycle corresponding to that. So in the case that the 3 cycle survives, right, the attractor certainly is affected by the addition of these peripheral neurons, right? So here, for example, we have identified, at least in the case of cycles, a way to isolate the relevant core periphery subgraph that'll tell us about what neurons are going to be active in our limit cycle. And so you can see that these particular peripheral neurons are picking up and firing at low rates. Okay? So the embedding is impacting the limit cycle and the sequence in that way. Okay? But what is it about the embedding that determines when the limit cycle actually survives? Okay? And it turns out, to understand that, we need to know a little bit more about how it is that these attractors are emerging from this system. Okay? And so the key thing is that we have these threshold linear dynamics really are piecewise linear. Okay? And so we can associate a hyperplane to each of these thresholded terms. So where the input is equal to 0. Because when the input is negative, this gets set to 0. Right? Where the input is positive, it's just itself. And so we can think about dropping this threshold nonlinearity as long as we know sort of which side of all of the hyperplanes we are on, so that we know whether we should set each term to 0 or whether we can just drop it and have a straight linear system. And so the point is that we get all of these different chambers of linear systems that are all pieced together somehow. And so here is an example of this patchwork of linear systems. So here are all the hyperplanes this is the 3 cycle graph in particular, and the hyperplanes cutting up the positive orthant for that. And the way that that limit cycle emerges for that 3 cycle is that it turns out there is an unstable fixed point that's a spiral out that has support 1, 2, 3, and what we mean by support is just that's the set of positive firing neurons of that fixed point. So the set of active neurons in the fixed point. So we have a full support unstable fixed point that is a spiral out, and as it spirals out, it loops into the chamber, a different chamber of linear system that then kicks it back into its first chamber. Okay? And then it loops back out, right? And that within the linear dynamics governing the system in this other chamber, push it back in. Okay? And so you can see that a little bit more clearly here. We've just colored the trajectory based on which chamber it is in. Okay? And so even though locally everywhere it's just a linear system, the patchwork of these linear systems creates these nonlinear dynamics. But really what's fundamental to shaping that was the presence of this unstable fixed point. Okay. And you can see that as well. So here is another example network where we have 4 overlapping 3 cycles around the outside. And we have an unstable fixed point supported on each of those 3 cycles. And we actually are getting a chaotic attractor for each one of those 3 cycles because each of these unstable fixed points has a stable direction in that corresponds to these sort of pinches as the trajectory follows that stable direction in, and then at random times it will end up spiraling out in these unstable directions. And so that's how we get these random pinchings and these chaotic attractors. But the key is that the unstable fixed points of each of these 3 cycles is what's shaping the corresponding attractors. Okay? And so in this sort of mystery networks, right, where we had Some 3 cycles gave us attractors and some didn't. The key was that the unstable fixed point corresponding to the 3 cycle 1, 4, 5 died in the full network. There was no fixed point there anymore, based on the way that 3 cycle was embedded in the graph. And so that's what we're going to get to next, is understanding a little bit more about the structure of the graph that's producing particular unstable fixed points and when those unstable fixed points will die. Okay. So one of the things that we found specifically in these networks is what we're calling this rule of thumb associating fixed points and attractors. So in particular, a core motif we're defining as any subgraph that has a unique fixed point where that fixed point has full support. So like that 3 cycle, there was a fixed point where neurons 1, 2, and 3 were all firing, and there were no other fixed points of the network. So that is an example of a core motif. And what we have found in just from computational analysis so far is this rule of thumb that every core motif has an associated attractor if and only if its fixed point survives in the full network. Okay? And moreover, you can access that attractor by just initializing your network at a perturbation of that fixed point. Okay. And these are, typically speaking, the only attractors that we see are ones that are arising from these core motifs. So just coming back to this first example where we saw all these different attractors of our network, this really was a CTLN, in fact, that we were running the dynamics for. And in this particular graph, the only core motifs are cycles and clicks. We will see that there are much more general things than that, but in this particular graph, they are just cycles and clicks. So I have listed for all of those for you down here. And we have survival rules or embedding rules for exactly when cycles and clicks will survive. Okay? And so all of these X'd out things are ones where we know there is no surviving fixed point based on that embedding. And I will tell you about those embedding rules in a second. And the bolded ones have a surviving fixed point, and those are precisely the things where we see a corresponding attractor. Okay. And in particular, I mean, this is one example, so don't feel convinced by one example. But we have done a complete analysis, as complete as we can hope for, in terms of needing to be able to sort of sample an infinite, an infinite space of initial conditions. But we did a very comprehensive search for all of the attractors of the 160 non-isomorphic graphs on n less than or equal to 5 nodes that are oriented with no sinks. And we were just interested in that family because that's where we could originally prove we had no stable fixed points, and so we knew we were going to get interesting attractors. And what we found there was that the rule of thumb, which says that you're going to have an attractor for a core motif if and only if the corresponding fixed point of that core motif survives in the full network. The rule of thumb correctly predicted the set of attractors for the full set of attractors for 154 of those 160 graphs for a particular set of parameter values that we chose. For 6 of those graphs, there was an extra core motif that did not have a corresponding attractor in that parameter range. But if we enabled ourselves to vary parameters, we could always find a corresponding attractor for the surviving core motifs. And so over varying parameters, that rule of thumb held for all 160 graphs. And one of the things that was interesting that came out of that is it just highlighted to us 5 additional core motifs that are oriented of size 5 that we hadn't necessarily thought about before. Okay, so this was just experimentally uncovering things that we can now prove are core motifs. So the 5 cycle, not surprising. The second one is a cyclically symmetric tournament, so each node has edges forward to the next 2 nodes. This one we're calling a cyclic union. I'll come back to it in a little bit, but you can think of it as like there's a single node, there's a 3 cycle where everybody's receiving from that guy, and then there's another node that receives from everyone in the 3 cycle and go back around. Okay. The other one is very close to that, but off by an edge, and then this last one is sort of a 4 cycle that's hopscotching through 5. Okay. All right, but the most important point of or the value of the rule of thumb was that we can actually connect fixed points to the graph structure directly through some beautiful linear algebra tricks. And so I'm not going to go into the linear algebra tricks, but I do want to highlight to you some of the graph rules that we've been able to develop to understand better these core motifs. Okay. So the first one is the one that covers the cycles and clicks, but also a number of other graphs. So we say that a graph has uniform in degree if all of the nodes, when you do an induced subgraph restricted to it, all of the nodes have the same in degree. Okay, and so here's just a sampling of such graphs. And we can prove that a uniform in degree graph always supports a fixed point in isolation. And we know exactly when that fixed point will survive to the full graph. Okay. So if each node has uniform in degree D, and when you embed that graph into some larger network, if you look at each external node in the network and check how many edges it receives from the uniform in degree guy, if any of those nodes receives D plus 1 or more edges, the fixed point will die. If each external node only receives at most D edges from our uniform in degree guy, then it survives. Okay. And so in particular, in the case of the 3 cycles, right, every node has in degree 1 in our 3 cycle. So if there's any external node that receives at least 2 edges from the 3 cycle, we know the fixed point will die, and there won't be a corresponding attractor. So just to recap, right, the fixed point survives precisely when no external node receives at least 2 edges, and that's what goes wrong with our 1, 4, 5 cycle here. Right? Node 2 receives 2 edges from that, and that is enough to guarantee that the fixed point of that restricted network will not survive to be a fixed point of the full network. This is just a quick blast of sort of all the rules that we have, or assortment of rules that we have come up with so far. We're calling things a permitted motif if they have a fixed point of full support in isolation, but we haven't necessarily ruled out that there are no sub-fixed points. So they aren't necessarily core. But uniform in degree is one rule that we have on that front. We also have some constructions for building new core motifs from old. So a disjoint union of permitted motifs will be permitted. It won't actually be core, but I'll talk about that in a second. A complete join, which we call just a click union because the skeleton is a click. But a complete join of core motifs will be core. And one of these cyclic union kind of things of core motifs will also be core. We also have some important ways of ruling things out as being core motifs. We termed this idea of graphical domination, which does not tragically align with the notion of dominating, of finding a dominating set or anything. But it is a way that we can guarantee that we won't have a fixed point support, and so that enables us to prove, for example, that directed clicks, if the directed click is not a full click, we're guaranteed that it is not a core motif. If you have a feed-forward network we are guaranteed it is not a core motif. We also know that your core motifs cannot contain sources, among other things. And we have a variety of survival and death rules for these 6 points. Okay. And one of the nice things that came out of that is that we could identify all of the core motifs up to size 4 and prove, in fact, that these are the only possible core motifs, or this is the complete set. And there are 9 of them. We also can prove that there are 37 parameter independent core motifs of size 5, and then 8 more where the existence of that fixed point actually does depend on parameters. And so it's one of those cases where we can't get away with just saying that this is something in terms of the graph. So just as I was mentioning before, one of the sort of most important things in terms of thinking about sequences has been the cyclic unions theorem that we were able to show, that if you have fixed point supports in each of these components, and then you are connecting your components by just all of the edges from nodes in one component to nodes in the next, then we know exactly what the fixed points are. They are just unions of fixed points from all the components. And so in particular, if each component had a unique fixed point, then this cyclic union also has a unique fixed point. And so a cyclic union of core motifs will generate for you new core motifs. So here are some examples of those. But one of the things that we found that's really cool is that a lot of graphs that are just one edge away, or in some cases even 2 edges away from a cyclic union, are also still core motifs, okay, and tend to have similar dynamics. So each of the things in this bottom row differs by 1 or 2 edges from a pure cyclic union, and yet still has similar dynamics and is itself a core motif. And so we know that this is telling us that there's probably some room in the proof that we had for the cyclic unions to generalize that sum. And we have been able to do one aspect of generalizing the inductive argument so that we can prove that if we use these guys as skeletons, so we inserted anything into each of these components, or inserted components into each of those nodes, then we would continue to get new core motifs built out of these. Okay? And so we can generalize somewhat from the original cyclic union results, but we still are looking for exactly what makes it tick to be able to generalize further. So now I want to just wrap up and go back to this original sort of graph that had some symmetry about node 2 maybe, but we really weren't sure where is this sequence coming from. And literally, Karina is my collaborator at Penn State. Her PhD student was studying this particular network like 3 years ago and found that it had this global attractor. And literally it was not until a week ago that we could finally understand with this theory exactly what should be happening here and why this makes sense. So this is all very recent results. But what we found was that if we use this rule of thumb now to analyze this graph, we can look for surviving core motifs. And we can prove that there are exactly 2 surviving core motifs in this network. One corresponds to this perfectly cyclically symmetric tournament on 5 nodes. And that was one of the things that was sort of symmetric about node 2. And the other corresponds to this cyclic union of point, 3 cycle point. Okay. And so that attractor that we were seeing, if we look at how that surviving core motif was embedded in that full network, we can see it sends 2 edges to node 2 from non-adjacent nodes, and 2 edges to node 5, but from adjacent nodes. Okay. And so we would expect that the neurons in the core motif are going to be the high-firing neurons, and that the peripheral guys are going to be low-firing. But one of the things that we have discovered is that an embedding rule or a core periphery rule for this particular motif is that the peripheral neuron only participates in the sequence if it receives from 2 adjacent nodes. Okay? And so node 2 is dying because it is not receiving from 2 adjacent nodes. All right. But interestingly, right, this was the only attractor for the particular parameters that we typically look in. But what about that other surviving core motif, right? We normally expect an attractor corresponding to that, right? And in particular, we could predict that it would have some kind of sequential structure like this, and that it should potentially pick up some of these peripheral neurons. And so we were surprised that we weren't seeing this in our standard parameters, but now that we had this understanding, we were like, all right, let's go search for it. And we were able to find a corresponding attractor for this. And so it turns out that we can, this sort of mysterious network that we have not understood for 3 years, we can finally make sense of with this philosophy, or this rule of thumb. So I just want to end there. I had thrown in extra slides that I knew I would not have time for about some symmetry stuff. But thank you so much for your attention.
Other question for Katie? So now you showed us some situations for rather small graphs, like 6 vertices, but you said you were like with the real situations with the 300.
Right.
How do you use your results as well?
So we are just now starting this collaboration where we are going to be looking for these motifs in large networks. And so one of our first steps is going to be to, we have some code to search for these motifs, but it is not going to be nearly efficient enough for that large of a network. So that's one of our steps. The other thing that's just really exciting about this new collaboration is that once we find these motifs, they actually have the ability now to optogenetically stimulate neurons in one of these that we have identified as part of one of these core motifs and actually see what happens after those neurons have been stimulated. And so it really would be like sort of initializing around that fixed point and seeing what happens with those dynamics. So there's extra potential for cool collaboration there that we're thrilled by. But,
Have you considered using retinal neural networks?
So, in the sense of things that inspired like the self-organizing that kind of stuff,
Well, I mean, the retina is part of the brain, basically, that's successfully been stimulated directly.
So the one thing is that sort of separates retinal a little bit from what we are thinking about is that because it is peripheral, right, and is responding to directly to stimulus, more of what we were interested in was higher up layers where it is not responding directly to stimulus, but rather that there's internally generated dynamics based on that connectivity. And so we specifically would not tend to look at that area just for that particular reason.
Can we postpone questions later? Otherwise we don't have time for time. Thank you very much.
Thank you.